want to they have progressed and they're promoting themselves, but how do they take the help from their neighbors? How do they take the help from their neighborhood environment? So let's look at that. And so it's just a small picture. Oh, sorry, I gotta share. So
Recording in progress.
This is a picture where you are seeing, you know, you are seeing a tumor that seems to be still contained, all right, but it has a microenvironment. It has cells around it. We don't know which ones are which, but it seems to have different, they are stained. There's different cells around this because I'm not sure what cells those are. I don't have that the diagram for this one, but just to show you that this is looks like it's fairly contained, or there are some cells that are uh, that are on the peripheries, etc., which constitute its microenvironment. So here, this text that I have for you, what it's saying is that the tumor cell can change the microenvironment, and then the microenvironment, all the neighboring cells, again will then affect also how the tumor grows. So they're like talking to each other. It's a combination of things. All right, so we're not going to go through these YouTube videos, but maybe at some point, if you have these slides, you can watch the YouTube videos yourself. So a little bit of basic terminology. And so the basic terminology is that in order to understand what I mean by microenvironment, TME, the microenvironment, what is in the microenvironment? To understand that, let's just look at these definitions. Parenchyma is a word that you might see here and there, and it refers to the uh, it refers to that part of the organ that's performing the functions. Okay, so epithelial cells, we just said, right? Epithelial cells, they are on the organ, they cover the organ. Epithelial cells, variety of epithelial cells, they make up different have different functions in the organ, and so these are it. So this is it. So that is called as the parenchyma. So so that's called as the parenchyma. On the other hand, the connective tissue around it, now I have drawn this here, like this connective tissue that surrounds all of those epithelial stuff. It's called the stroma. So the stroma could be anything like the nerves, okay, the nerves, the blood vessels, all right, that is supporting this. So that is called as the stroma, and they are in support. So they really do not live separately, they live together, as in this diagram. So the parenchyma and the stroma are really working together. There's just no way that the parenchyma can live without blood vessels and nerves and maybe other uh, extracellular matrix items. So what is an extracellular matrix? Anything that is outside the cell, outside the cell, outside the cell. So if there's anything that is outside the cell, all these things, these will be the extracellular matrix. And these would maybe all be the supporting cells. So all these supporting trophic tissues are called as the stroma. So stroma is like a big name for everything. If I say stromal cells, I mean everybody together. All right. And a glandular organ, again, the parenchyma can refer to the glandular organ. So exocrine glands, okay, a these cells, they secrete things, right? So exocrine glands, they are they are the salivary glands. They secrete salivary juices so that you can digest your food. Then the liver secretes enzymes to break down the fats. So they have exocrine glands. And the parenchyma can also be endocrine glands. Endocrine glands means that the thyroid dumps all its stuff, like the thyroxine, in the blood vessel. So it'll go in the blood vessel. Parathyroid, whatever it makes, goes in the blood vessel. adrenal glands makes adrenaline. It goes in the blood vessels. So endocrine glands, everything goes in the blood vessels. So this could be all for all of them. Now recall also, remember that we talked about this hallmarks, and hallmarks were saying that new things that they acquire, properties they acquire. So we said that cellular metabolism, acquiring this whole thing about glucose breakdown, a lot of glucose utilizing. So that's cellular metabolism, not having death. All right, so this is just for you to understand that all of these properties here, they are every time we are doing anything today, it's relating to these properties that the cancer cell wants to do. And so on the inside, if you can see, on the inside, if you take a look, then you have these cancers. Oops. On the inside, you have a blood vessel going. So this is a blood vessel. That's a blood vessel on the inside. And some of these are cancer cells. Some of these are supporting cells. I don't know what they are, but some of them might be different cells. Um, So a variety of cells are all together but joined by blood vessels. So blood vessels are going through them. This is just a reminder. So the tumor microenvironment, everything around the tumor, the different cells, everything, the tissue, the extracellular matrix, the nerves, the blood vessels, everything around the tissue there, around the tumor is now kind of like in a dynamic ecosystem. It's an ecosystem. They're all talking to each other. They're working with each other. So that's the ecosystem. And it is surrounding. So the TME, it surrounds the surrounding, and the TME is not only surrounding. So if this is the tumor, not only is it surrounding it, not only is the TME, so the TME is not only outside, but the TME is also infiltrating tumor tissue. So there's TME on in the inside because there's in between the cells. So it's everywhere. So it is the cancer cells also constitute the tumor microenvironment. You would think, well, why is the environment the cancer cells? But the cancer cells are also part of that environment because they're talking to each other. Then there are supporting cells, like we'll see what they are. And then there's the ECM, the extracellular matrix. The extracellular matrix is like a ground tissue. It's like a ground tissue with proteins. So it's a ground tissue with proteins, and one of the important proteins that it has is collagen. Okay, so collagen is an important protein. Collagen is the same thing that you find in meats. When you cook meat and then you leave it to cool and then you eat the next day, it becomes like a jelly, so that has all that collagen in it. And together, and signaling factors, signaling little chemicals that are going back and forth between cells in the outside. And so together they govern their behavior and progression. So let's look at that. So important components of the TME, the tumor microenvironment. I have a picture very soon. So let's look at this. So first things is that it has to have the tumor cells, okay? Then it'll have these infiltrating cells. It'll have other resident cells. So cells, cells, cells. It's a lot of cells that are in the TME. And so and then it'll have substances that are secreted. And then we are not going to talk so much about hypoxia here, but yes, we will talk about a little bit more about angiogenesis. And we're not talking about this one. So let's look at all of this together. It is looking like it's kind of like a few couple of cells, but it's really really, really complex. Okay. So I try to make a big, big topic in somewhat simplistic way for you, but it's just not easy. So here it is. Um, so some of these slides will have a lot of things written in it just so that later on you can go back to it. So composition of the TME is complex, and again, it's a ecosystem. So the cancer cells, the stromal cells, all the cells are together, all the cells are immune cells, and then the ECM. So this is just a breakup of this simple slide here. Okay, so this is just broken down if you wanted to read some more. It's just in more detail, but we're going to cover each of them. Okay, and with all of these, so if you look at the breakdown, it has cells, cells, and cells. Then it has soluble factors. soluble substances. Then I said it has ECM, extracellular matrix substances like collagen, and then it has blood vessels and neurons. So with all of those things, which is all right here, everything is right here, and it has the stromal cells. Stromer cells are all the surrounding cells. Um, then with all that, what is its function? What is the total function? Well, the total function of the tumor microenvironment is to support a full biomechanical support to the tumor, full support to the tumor for its growth. Biomechanical. So did you hear the word mechanical? That means there is substances in there, or they have to be things in there that help it to be stable mechanically also. Okay, so for mechanical, for growth. And so the ECM, the extracellular matrix, what is the extracellular matrix? Every time you have a cell, on the outside is extracellular matrix. It is sitting in it, okay? So that extracellular matrix influences all of that, makes it stiff, gives it surface structure, all right, and it gives signaling, okay? And you can ignore the rest. So what are the other things that it does? So biomechanical support through, you know, physical support really. Then immune modulation. That means with the tumor, the immune cells around the tumor, all the immune cells are around the tumor. Okay, I have a picture coming soon. Um, all those immune cells are backing off. The tumor is saying, don't come and get me. All right. I'm the good guy. Don't worry. I won't do anything. That's it. So all the immune cells, the T cells, the B cells, the macrophages, whatever, they are not going out there to attack this tumor. So it is evading immune surveillance. So immune modulation. Okay, it's explaining to them to stay put. And then it is remodeling everything. It is remodeling everything around it. And it's saying that I want this furniture here, I want that furniture there, I want the collagen here, I don't want the collagen there. It's remodeling everything. And the tumor is saying, I want things where I want it because I can grow better. And then the tumor says, well, I need to make blood vessels. So, blood vessels, can you please help me? And here it is. This is what you want. You want this signal VGF, vascular endothelial growth factor. I give it to you. I'll make VGF and I'll give it to you. Please give me some more blood vessels. This is it. Metabolic reprogramming. It says the cells are going to multiply a lot, and they're going to use their glucose in an efficient manner. Okay, and this is how it will all look like. So to put it in perspective, um, to put it in perspective, here's a existing blood vessel, and the existing blood vessel has branched out. It has branched out. I hope you can see this. The existing blood vessel has branched out and it has gone inside the tumor. So that is, sorry, that is, it has branched out and it has gone inside the tumor, and it is providing it oxygen. It is providing it, it's not taking all the oxygen, but it's providing it some nutrients through the blood, etc. And it's taking away the waste. And so what else do we have here? We have all the cancer cells. So those are the cancer cells. Then we have some macrophages. These are macrophages. Macrophages are like they're like your immune cells. They go and they go swallow cells, but in this case they're like tolerating everything. So the macrophages, they will supposed to go up and swallow these abnormal cells, but they're not doing that. So well, the only one that I want you to look at here is the this one, the fibroblast. So that is the fibroblasts. So there are these fibroblasts that are, and so pay attention that the fibroblasts, not all of them are on the inside. There's only one here on the inside. All the others are towards the outside. You see that? They're all towards the outside. And so these fibroblasts are their connective tissue. They secrete things. They secrete substances. And they will be careful. They will secrete it if it helps the tumor for some time. And they will, so the this tumor is telling the fibroblast to become a good friend and to secrete only things that are useful. So the fibroblast is going to is being converted into a friendly fibroblast that will only help. Okay. And other cells are pericytes. Let's look at this very soon. And you have your T lymphocytes that are supposed to go and kill the tumor cell, but no, they are just happy sitting here. Okay. And I want you to look at these little things, these little dots here everywhere. So here's your T lymphocytes, not really doing much. Oh, other immune cells, but not doing much. All right, or trying to do things. And here I want you to see these. These are cytokines. Cytokines are substances that the cells secrete also to talk to each other, to for inflammation, for anti-inflammation, etc. So that whole is the tumor microenvironment. So the tumor microenvironment is not on the outside. It is not just here. It is not just these. The whole thing together, even whatever is inside, if there are cells that are on the inside, then that is also the tumor microenvironment. Okay, so everything that's also in here. And you have some pericytes. Let me just take you to another slide where we can talk about that. And so here it is. So your neoplasm, you don't have to pay attention to the words anymore now that you know the growth of this tumor. You know, it was a neoplasm, it's just a couple of small cells. So the growth of this tumor depends on how well it uses all the resources in the tumor microenvironment. And if necessary, it can also undergo regression if the tumor microenvironment is kind of taken away from it. No, no, it's not a happy tumor microenvironment. So that's also possible. So, so now take a look at this. The parenchyma, which is the parenchyma, are those cancer cells. They have turned cancer. So that is a neoplastic parenchyma. All right. So what is inside it? Then there is the stroma. This is the cells that secrete, and then there's the stroma. Stroma is non-neoplastic. They are all, none of them are cancerous. So the stroma, all those cells, none of them are cancerous. Only the cancer cell is cancerous. And so you don't have to pay attention to which one is what, but see, look at all the things that they have. So all the stroma, that means the blood vessels, etc., it's providing oxygen, okay, whatever it needs, and it's providing nutrients. And so tumor growth, and so first of all, take a look at what these are. So you have, of course, you have your cancer cell, you have a couple of normal cells. Then you have these fibroblasts. Remember I said fibroblast? They have become now friends of the cancer cell. So they are cancer-associated fibroblasts. So they are CAFs. So some of them are trying to help and some of them are not trying to help. So they are cancer, they're associated with cancer. So they are cancer-associated fibroblasts. Then you have the extracellular matrix. Because why? Because you need collagen here and there. So these are probably collagen. So collagen fibers. And of course there's blood vessels. And this one we will talk about this I think a little bit today. Is a macrophage. What's a macrophage? A macrophage is a cell that goes and devours, eats up another cell, just completely engulfs it. And so this macrophage here, instead of it's wondering what to do, it wants to get rid of the tumor cell. Maybe it does, maybe it doesn't. So it's associated with the tumor. And so it's called as a TAM. So, so tumor associated macrophages are there. So those are the cells, and so tumor growth will go up, will be increased, you know, with the help of CAFs, with the help of cancer-associated fibroblasts. The tumor will grow better if the TAM, the tumor-associated macrophage, also says, "Okay, I won't, you know, engulf you." And so that's just at the immune level, and we'll just keep it at that, okay? Because the others are too complicated. Okay, so we have your cancer cells, then we have some cancer stem cells, and we have tumors with diverse phenotypes. Okay, so what does that mean? So, uh, so tumors with diverse phenotypes. So what it is, what we're trying to say is that everybody kind of like works together here. So, heterogeneous, the tumor itself is heterogeneous. Okay, the tumor is heterogeneous. That means it has uh type 1, maybe it has one type of tumor cells, two types, a second type of tumor cells, and third. And let's say that this is, I'm just making it up, let's say that this is colorectal cancer because we just did it. So all of them are not from the same clone. Some of them are the gray type, these are from a different clone. Some of these are slightly different, they have, and maybe this one has a slightly different mutation. So a heterogeneity in these tumors is a problem because now you have all of these working together. All right, some of them have mutation in here, some of them have a different mutation, and they're all working together. And there are some normal cells who don't know, who are still trying to figure it out. So a heterogeneous tumor, all of them together are working in cooperativity, and so they are so how are they working? How do we know this? Well, the way that was done is, you can ignore what this is saying. The way that was done is we separated this clone A, and then we found that it formed a tumor, but it did not make blood vessels. Okay, it did not make blood vessels, and it cannot invade. Then we took number two, okay, and we grew it separately. All right. And we grew it separately, and it's making some substances, and it's working by itself. And so it made blood vessels, but it cannot invade. All right. And then we took number three, and when we grew number three, and we found that, oh well, it does keep on forming a clump, okay, of cells, but it does not make blood vessels, okay, lacks blood vessels. So this is called as tumor heterogeneity. It seems to be they're all colorectal cancer, but somehow some of them just individually survived, and they are here in this mix. And therefore, because of this mix, this tumor kind of like is cooperating and working together. So where were we? We were saying that tumors with diverse phenotypes, diverse activities. Okay. Now, cancer stem cells. So one is cancer. What else do they have? Cancer stem cells. Stem cells, you must have heard, they are cells that they can become anything they want to, right? That's what we know. Stem cells. That stem cells can become, if I give it the right trigger, then they will become anything they want to. So we found that now this tumor has cancer stem cells, and that's kind of scary. So they are found in these solid tumors. They need like a solid tumor, and so they're very rare. There's only few of them because stem cells are always few. They're never too many. And so these stem cells are very small in that population. But what they do is, a stem cell has the capability to self-renew. What that means is that this stem cell in this whole population, so let's say that there is just one stem cell here. Let's say that this one that I just covered is a cancer stem cell. Then this one cell will make a clone of itself. It's like me. I make a clone of me. I don't give rise to my daughter who looks, who my child who may look a little like me and who look like me and maybe a little bit different. I give I give rise to a clone, which is just me. So a cancer stem cell, when it multiplies, it will be exactly the same as if it was brand new and young. So if I multiply, I'm 30 years old, I'm going to make a clone which will be less than 30 years old. Okay? It'll, so every time it's a clone and it renews. So that is called as renewal. That's called as self-renewal. Self-renewal means um, it's not getting old. It just made a clone of itself, and it's ready to start all over again. So that's a stem cell. I don't know how to explain it in any more simpler words. So, and the second thing about the stem cell, cancer stem cell, or any stem cell, is that it can differentiate into multiple cell types. Ideally, a stem cell, if I give it the right trigger, it can become a neuron, it can become a skin cell, it can become a heart cell, It can become anything. Uh, so so that's a cancerous stem cell, and that is there. Why is it there? That's scary, but the fact is it's still there. Okay. And so we don't know why, but that means it will make the cancer tumor, it will make the tumor, survive better. If the tumor is not doing well, it'll just make some more. The stem cell will make some more. The next thing um, here is these cancer stem cells. Okay, I'm just going to go through something. And so, one major property for all of these cells that we just talked about right now is that they're all epithelial cells, right? We said that most cancers, solid tumors, are all from epithelial cells. It so happens that these epithelial cells, they can convert into change their physiology. They can change their physiology, and they can acquire a fibroblast like phenotype. So what is a fibroblast like phenotype? So this is a cancer cell. It can become like a mesenchymal cell, and it can look like this. And so what's the advantage of looking like a fibroblast? It can try to sneak through the blood vessel. So it can travel, so it can migrate, it can sneak through the blood vessel, and it can go inside the blood vessel, and it can come out. So that's a fibroblast phenotype, and then when it comes out, it becomes like this back again. So this is called as epithelial mesenchymal transition, EMT, and we're going to cover this later on in another lecture. And so that's also one of these tumor environment cells, the tumor cells, the cancer cells, they will do this. They can have this property. Okay. So the next is these lymphocytes and macrophages. So just a small thing about these lymphocytes and macrophages. So what are we trying to say here? These are lymphocytes are cells that are supposed to um, either go at target and kill cells, like touch them and kill them. Um, and macrophages are supposed to go and kind of like travel and engulf them. So macrophage, that's why the macrophage kind of like looks like it's shown the way that it is. So a macrophage, a tale of two roles instead of a tale of two cities. There are a tale of two roles. Macrophages can be of various types. The same macrophage today can become a supporter of of one leader of one type of cell, and the same macrophage tomorrow can become anti. So this macrophage is just going into politics. That's all. So these macrophages, when they are in M2, the name when they are in M2, and honestly they look the same. So this macrophage, when it is in M2, then it is pro-tumorigenic. It supports the tumor. How does it support? By not doing anything. By not doing anything, by not secreting anything to make the cell unhappy. So where's the cancer cell? Here's your pink cancer cell, right? I think so. So here's your pink cancer cells. And the M2 is just surrounding it and kind of like helping. Okay? Just helping it. So the macrophage on one day because of certain signals decided that it's going to become a helper. It's going to kind of like support and not do anything bad. Is that okay? So it is pro-tumorigenic. On another day, by a different signal, the same macrophage can become the same one or maybe a different one. So maybe they're two different populations, but we're hoping that they can interconvert. That's what we hope, that they can convert maybe. So left arrow, right arrow. And this macrophage can secrete substances, or it can go and kind of like engulf, and it can try to kill the tumor. So that is anti-tumorigenic. And that's the video of these. That's just the story of these macrophages. So macrophages, depending on, we don't know why the macrophages today will support and tomorrow will be tumoricidal. It will destroy. It will try to destroy the tumor. So we don't know why they do that. We're trying to understand why the same macrophage can think of doing this. Okay, so you can ignore the remaining. And so what else did we have? Lymphocytes. The lymphocytes may not do much. So these are cells that they infiltrate here, and then they're just waiting and watching. They're waiting and
Microenvironment, or
watching and trying to
TME,
help.
refers to the
Okay. So at this point, I will show you this. So let's try to put it all together for you.
The tumor microenvironment, or TME, refers to the complex network of cells and structures that surround and interact with cancer cells in a tumor. The TME is comprised of many different cell types, including cancer cells, a wide variety of immune cells, and cells involved in structural support like stromal cells. In the TME, cancer cells can manipulate and hijack normal cell function to better serve their purpose, allowing cancer cells to survive and multiply. One major activity of cancer cells is evading the immune system and promoting an immunosuppressive TME. The immune system is the body's first line of defense and typically detects cancerous or mutated cells and targets them for elimination. However, cancer cells have mechanisms to evade or suppress the immune response, essentially making the cancer invisible to the immune system and allowing cancer cells to grow without restraint. Researchers at Bristol Myers Squibb are working to understand how the cellular components within the TME interact in order to discover and develop novel therapies designed to overcome key immunosuppressive barriers. This includes targeting cancer cells to make them more visible and more susceptible to immune cell attack, and directly modulating immune and stromal cells to optimize the body's ability to mount an effective anti-cancer immune response. Bristol Myers Squibb is dedicated to developing new therapies and exploring new therapeutic combinations that are informed by the complexity of the TME in order to advance the next generation of cancer therapies for patients.
So I hope you understood that what we would like, not for you, but for my drug design, this is what I'd like to do,
Interact in
is
order to
that
discover and develop novel therapies.
is that the cancer cell can be treated in such a way that these immune cells, these macrophages that were staying away, will now recognize it and will go and start doing their work. So that's what the immune cells need to go back and do. And so that whole tumor microenvironment, which is beginning to support it's beginning to hijack the whole system, that support will not be there anymore. Okay, so let's go back to our. So then was the fibroblasts, and the fibroblasts are cells that are support cells. They're there anyway. They're around here in these tissues in various places. Endothelial cells are for blood vessel. This is for blood vessel. Adipocytes is for fat. Microbiome is, well, in our GI tract, we have bacteria. We don't have bacteria everywhere, but in our GI tract. And so fibroblasts, okay, so fibroblast and fibroblasts are now have been kind of like primed to become cancer-associated fibroblasts. All right, so we find that all of these changes in those cancer cells, they have all these tumor suppressor mutations, etc. They that they are also found in that stroma, and so they are making the changes. The cancer cell is making the changes in these other fibroblasts. That's what we're trying to say. So these are your fibroblasts. They look like this, remember? And so now they're cancer associated. So anything that's associated with the cancer, it's trying to tell it to work for the cancer cell. And so they produce collagen, and generally they produce collagen, but now they're going to maybe produce less collagen, or maybe they'll produce more collagen. Okay. And they will make these proteases to break the proteins. And so these CAFs can now promote tumor cell invasion. So they will make these proteases break the proteins, and then they can go anywhere and they can remodel the ECM, remodel the ECM, remodel the surroundings. That's what it means. So CAF is now becoming a nice worker for the tumor. And remember, it makes collagen. So, okay, it produces collagen. So maybe now it will produce less collagen or more collagen. I don't know. Let's look at it. And cancer associated mesenchymal stem cells. Okay, so these are a subset. Uh, these are a subset of these fibroblasts. Fibroblasts, again, they are connective tissue. And these, because it's mesenchymal stem cells, they can become anything they want to. They can become fibroblasts, they can become endothelial cells. So essentially, those are the cancer stem cells, or a type of cancer stem cells. So they can become bone cells, they can become other things, depending on what the, cancer cell wants, cancer tumor wants. And so they promote progression and metastasis. And we found that they are particularly important for ovarian, colon, and pancreatic cancers. Okay. And this is just to give you a summary that this cancer associated fibroblasts that I talked about here, that they work for various, to promote and blood vessels, etc. So they will they will help support, okay? They will help support the whole thing by secreting these different chemicals. So they will they will secrete these different chemicals in order to allow proliferation. They will secrete these different chemicals, a very important one being VEGF, vascular endothelial growth factor, to promote blood vessels, more blood vessels form. They will secrete all of these substances such that these macrophages just don't do anything. Okay, they just don't do any work as they're supposed to do. They're quiet. So immune suppression. All right, and the last one here is that they produce collagen 1, collagen 3, things like that. Collagen is all these fibers here. And if you make a lot of collagen fibers, then the drugs cannot pass through. So the fibroblasts will decide how much of collagen they want. If you make less collagen fibers, then maybe the cancer cells can pass through. So ECM remodeling, that's what this means. So I hope this picture gave you the full idea. And I think almost towards the last of these cells is endothelial cells. That's for generating the blood vessels. So endothelial cells are also part of the stroma now, and they can help by creating abnormal tumor vasculature. Abnormal, so they can help by making more vessels. All right, and pericytes, they are actually a little bit surrounding. They're smooth muscle-like cells, okay? I know that it doesn't mean much to you, but they surround the capillaries. They surround the capillaries and they help in that cell survival. They help in the uh, endothelial cell survival. So essentially, they stabilize the blood vessels. So they stabilize pericytes. They will stabilize the new blood vessels of the tumor. Okay. So essentially, they're just supporting blood vessel maturation. All right. So that's your pericyte. And where did you see a pericyte? I think we saw one here way early on. Let's see. Did we see pericytes here? We saw some pericytes here. There you go. So we saw some pericytes. That's a pericyte. That's a pericyte. And see, the pericyte is going to go and travel along, and it's going to help support the blood vessel, and so that it stays. So that's a lot of cells. Do you see? It takes a whole village for a tumor to go. And here's one tumor that is hijacking the whole village and telling it to work for itself. And so here's a little picture for you to show how that happens. So VEGF, that is a very important growth factor, vascular endothelial growth factor. The cancer cells will start to make that. How does it make that? Because first of all, the tumor becomes hypoxia for some strange reason. It has hypoxia, so then it makes vascular endothelial growth factor. It uses glucose, and then it starts sprouting the blood vessel. Sprouting, that's the word, sprouting capillaries. And so here it is. This has become endothelial cells are being made on the inside here. Endothelial cells are on the inside, and it makes this blood vessel, and it keeps on making more blood vessels. And so now this is why you see it looks like a Kankara, like a crab. And so that's your pericytes. The pericytes are helping the process. And so further metastatic spread and growth of the tumor. So cells of the TME really help in vascularization. And another similar one, but kind of um, just so that I could just give you this picture of a tumor. Actually, it's a cartoon on top of a real picture. Okay. And so just to show you how vascular angiogenesis occurs. So this is how angiogenesis occurs. Angiogenesis occurs by forming little tube-like structures and then branching and further branching. All right. So and so here's the cells there. The cells are on the inside. This is your endothelial cells, and it just forms more branches. So it's called a sprouting. That's just the name. And so now I will show you a video of angiogenesis. So this is a blood vessel, and you see it's just like I showed you in the schematic, it's leading out these tube-like structures. First, they're open tubed, and then they will start to join in, and they will start forming up blood vessels further. So this is on a time-lapse video, and it starts to branch and branch and further branch out. So here's and that's your blood vessel. Look at all those endothelial cells that are lining it. And then VEGF comes along. Where does VEGF come along? It comes along from the tumor cell that starts to secrete it. And so one endothelial cell will now start to begin to sprout. So this is the literal scientific word is sprouting. So sprouting, there you go. And it needs to, this is just a cell, there's nothing here. It has not formed a vessel yet, but now it will start to form, it'll start to reorganize its center, and it'll form a tube. There you go. It forms a tube, and it forms this lumen structure, and that's where new blood will go in. So this is a endothelial cells. Look at them. They're just two separate cells separated out. They're joining, and you can see how it's beginning to form, joining together, and it has formed a lumen. It's amazing how this can be done. And that's a fluorescence microscopy to show this. So next. So if you have a tumor that is now secreting VEGF, it's VEGF or VEGF, it's secreting all of this, then some of them, see the blood vessel on the top there, that green blood vessel that's on the top, that somewhere along the line, it will start to make a branch and it will reach out. And that's how it's doing it. So even though the tumor is far away, it is secreting these VEGF such that branching starts to take place. And that's how it has reached out. So remember, blood vessels can only form from an existing blood vessel, not otherwise. Okay? Alrighty. Let's go back to. So I hope you liked that video. And lastly, it says that there's, I'm not so interested in this, but adipose tissue. That's like fat tissue. Adipose tissue means underneath your skin you have fat cells. And so these adipose tissue, I cannot call them fat cells, but you know, understand what I mean. So they are responsible for some changes in metabolism. So again, if the adipose tissue, if the cells are lean, all right, then they have one kind of role, and if they are obese, then they have another kind of a role. So if they're obese, then they will invite other cells, and then all kinds of they will secrete all kinds of stuff in order to activate cell proliferation. So all I just wanted to say is that lean cell versus obese cell allows for changes in the surrounding stromal cells again, such that it allows for it to new signals are released, and it'll promote tumor. So it's pro-tumorigenic. And so where is this really common? Those fat kind of cells that we're just talking about. So adipose tissue when metastasis occurs. So in metastatic site for ovarian cancer cells, all right, that is uh, so ovarian cancer cells, they like to go into the belly, into that fat tissue, and they like to go and work in the fat tissue. And here we found that, you don't have to go through these slides, but we did find that the fat tissue, adipose tissue, the marker for the adipose to the obese, adipocyte. The marker is FAB4, and it was found high at the metastatic sites. And that simply means that it just prefers, and metastasis is kind of preferred for places that have a lot of adipose tissue. So, and lastly, I don't know much to talk about this right now, but the microbiome, this is a big thing now. The big thing in this last 3-4 years is the microbiome. What is the microbiome? In your GI tract, here, only in your small and large intestine, you have lots of bacteria. And so some of those bacteria are they these are the intestinal microbiome or microbiota, and they do some signals. They make your, sorry, not stomach, they make your colon secrete things. So a lot of our secretions happen in the colon, where it is happening by the bacteria, by the gut bacteria. And so all of these gut bacteria, these are the names, you don't have to go through them. They are responsible for in you know there's responsible for, if they're over infected, then they can be responsible for a chronic, they can be responsible for chronic inflammation. Now that's not a good thing. Okay. So if there are toxins, if you have toxins from a certain bacteria, then also they can, cause hepatotoxicity. But the reverse, which is not written here, the reverse is these days is being studied, is that if you have good bacteria in your GI tract, then there are certain diseases that you will never get. And so that's also another thing. Here it is blaming the bacteria for all of that, but it is also the opposite. And also in recent studies is there, where the cancer cells, hopefully, and other diseases, like for example, your gut bacteria and asthma. Okay, just going off topic for a bit. Asthma has been found to be controlled if there's the right type of bacteria in your GI tract. Let's say you eat yogurt every day. I'm just making it up. Then you have a good quality bacteria in your GI tract. It has been found that even as things like asthma are also related to that. So a lot of diseases can happen or not happen because of this. Okay. Lastly, let's look at these soluble factors, and I really need to explain some of, well, just to overview that these are soluble factors. Cytokines and chemokines, these are the names. All right, we said just VEGF. We said matrix metalloproteins, MMPs. They go chew up the ECM. So these are all required. And so essentially, all these cytokines, they're just released. They're substances that are released. These are chemicals that are released, and they are telling the breast cells to do things, or they are allowing the cells to multiply or to travel, etc. So these soluble factors, all right, they help. So if there's too much of these soluble factors, they can help in metastasis also. So let's go to the next one. So now here's something really, really different. This is something really different. What has happening here is that the the tumor itself has regions of different types. The the regions on the tumor itself, the different regions are heterogeneous. Just because all the tumor cells are big lump doesn't mean that at any point you take a biopsy and you'll get a snapshot of everything. No, that's not true. What is happening is that the probably the blood flow is not very good on the core inside. On the core inside, the blood flow is not very good. So the heterogeneity of the blood flow, all right, contributes to acute and chronic hypoxia in tumors. So we just said that tumors have hypoxia. They don't like to use the oxygen. Um, and so it could be acute. So where would it be acute? It would be acute in the center. So if it's acute in the center, then those cells kind of behave different. So how do you look at this? This is necrotic, means it's kind of like really dying tissue. But there's a lot of pressure. There's barely any oxygen, no PO2, no oxygen pressure. And its pH is kind of normal, but there's no proliferation. And it may or may not take the drug delivery. And this side, the outside, is really the proliferative one. The outside is the one where the cells are proliferating, means multiplying a lot. And this part is quiet. This part is quiet. So it's quiescent. This part is quiescent. So it's kind of very, very new, and not new. It's still a lot of research is being done about the heterogeneity, not only of the different tumor types, but the whole tumor as a body. And so and therefore getting different oxygen, etc., then will mean that it has different pHs, etc., and it has different conditions, and so therefore that leads to resistance to therapy. So this tumor will not really act in the same way. And so again, we said, why is it? It's only because of, especially because of the oxygen, and also solid stress. There's too much stress here. There's vessel compression. All right. So heterogeneity of this blood flow leads to this different pHs. So these different pH here, this different pH, and different conditions lead to the fact that it is resistant. And so just remember that ultimately hypoxia has a long, long goal. This hypoxia inside the tumor can even drive the cell to metastasis. All right, so the cell will now is acting, this tumor is getting really much more stronger and more metastatic because of hypoxia. So everything really is, we can put the whole blame on hypoxia. All right, and these are just ways of pictures of way to show you the same thing in various ways. All right, so all we're saying, this is a summary in totality. All that I'm saying is that the TME actually initiates, the TME really starts the metastatic pathway. So because of TME, the cells will now, cancer cells will go into the blood, they will go into the circulation, and then they will come out of circulation, which we will look at tomorrow. and then they will go and they will sit at another place. Remember, it's not always successful, but sometimes it will be. There will be a little bit of metastasis, and then it will be big time metastasis. So the tumor microenvironment seems to be the beginning of the metastasis. And so how would you study this? You would study this again. You can use a slide to mount stuff on cells on, and you can stain it with various stains and perform immunohistochemistry to be able to get either by regular bright field detection or by fluorescence microscopy. Both sides you can do this, okay? And this side is really for DNA. Um, this is more for DNA work, but this side is more for cytochemistry. And so you can again stain it with whichever things that you feel right. But this is really it, just fluorescence or bright field, not much. And again, anything that's blue here is normal. I mean, is not what you're looking for, and the brown one is what you're looking for. So the brown stains in these are the protein is what you're looking for. That's the protein that you want to look for. Further, we're not going to talk about much of this, that if you had a bulk tumor sample with all these mixed stuff and the stroma and everything, then you can remove all the dead cells, and then you can mix it all up, and you can analyze the type of cells by something called as flow cytometry, and you can do a genetic analysis both. So everybody does both actually. Every time you do a gene analysis and you do a flow cytometry. All right, this looks like a lot of writing here, but I just wanted to show you that this is the section where we look at slides, where we look at cytology. And so I've made this for you. So this is a machine learning project that was done back in 2015. So if you see at the bottom here, 2015, I took it from that paper, and they quantified the TME composition, all the cells in the TME. I showed you about, you know, 10, 15 different kinds of cells. And they did this in the ovarian cancer with H&E stain. Remember the H&E stain from yesterday? And what they did was also get this. So they look at the top. So they were able to, this is all, they took the, um, they're just showing you, that these are the cancer cells. They stained them, and then they trained the machine with 900 cells. No, they annotated. So, oh, sorry, 900 cells were annotated. Yeah, and from which they, they quantified these 100 morphological features, 100 features they qualified. So not just 10, 15 features, 100 features they quantified in this particular project. And why those 100 features? Because they're looking at epithelial cells, stromal cells, they're looking at fibroblasts, they're looking at everything. They're looking at blood vessels, they're looking at lymphocytes. So obviously you will have so many features. And to make a long story short, they looked at all of these features here, and what they did was then they classified them. Onto epithelial like green, and spindle is red, and the lymphocytes are blue. And they went on to see if they could kind of like automate this. And I can't remember what the results were, but the bottom line is that it was done for 100 features for over about 20 different kinds of cells. And so this is just for their, I think they did the validation work basically on this kind of tumors. That validation work was on the tumors, where then they stained them, and then they kind of like matched it up with what they got. And so this is really staining, and this is what is it called? The spatial distribution is showed here, and cancer cells are 70%, stromal are 19, and 10% are lymphocyte. Okay, and I think they have two different samples here where they just showed you again cancer here is 50%. Stromal cells are also 50%, and about lymphocytes are about 10%. So it is the whole um, by cytology, this could be done, and the whole thing, the whole tumor could also be examined. And so let me talk about some more modern stuff. Again, it's not that modern because it's again from 2014, but you can integrate the histology and the genome. So I'm not going to walk you through this whole slide, but you can integrate not just, you can take one slide and you can do the histology and you can do the genomics. You can take the DNA from there and you can study which kinds of, and get some kind of learning, some kind of learning, and have predictive models to determine whether what kind of cancer it is. And this is the same thing. These are from the same paper. All right, these are the same thing. All I'm just showing you here is that they were able to do by genome analysis, they were able to also, validate, and they were able to have more information as a data set. So they had genome information, and then they examined these digitized images, and they found that the tumors from the mesenchymal gene expression class, they are combining gene data. Can you imagine? They're combining all the gene data. They're still doing it today, combining it, and they found that the tumors are enriched with necrosis. There's cell death in those, okay, with mesenchymal genes. So, so then they found that certain gene expression makes it hypoxic in certain regions. And so I'm not here to talk about their study. I'm just here to tell you that these regions could be identified, and they are combined with genomic studies. So now I'm going to take a slight turn for something that is kind of different. And this is also cytology, but a modern twist to cytology. It's a paper from last year. And in this paper, they looked at ECM, and so they're looking at the ECM. What is ECM again? If so, this is your tumor. This is your tumor. Okay, let's say this is your tumor. Then ECM is everything that is kind of like outside here. All right, so it has fibers. It has this is the extracellular matrix, ECM. That's the matrix. So collagen is the most important fibrous protein, right? So they look like this. They look like this. This is collagen in the ECM. And what does collagen do? It is resisting tensile force. It gives structure. It gives structure so that the cells kind of stay in put. They don't move around everywhere. And collagen, just for your knowledge, is the most abundant protein in mammals, about 25%. So because it's not from your muscle, you think it's all from your muscle, but collagen is between them. Collagen is in between the cells. So 25% protein mass. Anyway, that's just a side story. What is the important story that we're trying to tell you? What we're trying to tell you is that in normal cells and cancer cells, what's the story about collagen, and how can I do cytology or histology for collagen, and how can I turn it into some kind of a machine learning experience? Okay, this is fairly new. You just need to think about it this way. So in a normal ECM, a normal cell, where is collagen? What is it like? So the collagen that they're showing you is here, like here. So this is the collagen, but the collagen is, yeah, that's your collagen, okay? And so if you look at it, it's like really big bundles. So your collagens are going left, right. They're just big, big, big fibers. They're holding your cells in place. Okay, they're outside the cells. So this is a normal cell. Sorry, this is a normal situation, and that's your. This is the ECM. So this is all the ECM, and these are the cells. The cells are in the ECM, and where is collagen? Collagen is in the ECM. Okay. Now look. So in these normal cells, I think this is normal. Yeah. So you see all these collagen fibers. You see these collagen fibers. Anything that's light blue, that's it. And these are the fibroblasts. This is the fibroblast. That's the fibroblasts. All right. These are the fibroblasts. And you don't need to know the rest. Okay, so this is all skin cells. So these are five. So all of this is collagen. Now, um, look at look at here. You can just see these collagen fibers. You can see these collagen fibers. They just look pink. They don't look any fancy to me, but they just look like there's like a lot of collagen fibers. Is that okay? So collagen typically looks like this. If you have a diagram, it's a triple helix. One, two, three, one, two, three. It's a sorry, one, two, three. It's a triple helix. So at the if you ever you see a diagram, it's a triple helix that just collagen. Three. So now let's see, where do the collagens go? Where are they made? The collagen is made inside the cell. Okay, so the fibroblast makes the collagen. Where's the fibroblast? Well, the fibroblast was not was here. Let's see. So where's the fibroblast here? That's your fibroblast. This will make the fibro collagen. It will secrete it. Look at this huge collagen, but it's made and secreted. So let's go to the next slide. So here's your cell. This is inside the cell. It will make these small collagen fibers and it'll throw it outside. It so this is your collagen. It'll throw it outside and then all of them will get together and they'll have these striations. So collagen has these striations. And then they'll make big fibers. And when they need to be broken, then they will go back. They can come back into the cell. They will be taken into the cell and they go inside the cell and then they get broken and then they get degraded. And that's how they're degraded. So two things are happening here. Okay, it got synthesized and it got degraded. So who's making the collagen? Well, of course, cancer-associated fibroblasts. CAF is making the collagen. If CAF is making the collagen, then maybe it wants to make it too much or too little. I don't know. All right, so cancer-associated CAFs they themselves are heterogeneous. Okay, they're heterogeneous, but they're not like your normal fibroblasts, but they're a little bit different. And so it is secreted by these CAFs. So collagen is going to be secreted by the CAFs. Do other cells make collagen? Of course, yes. All right, of course, other cells will make collagen. Okay, so but CAFs will, um, cancer-associated fibroblasts will. All right, so others do, but CAFs also do that. Okay, so they'll make the collagen. So let's see, should they make too much collagen or should they make too little collagen? And how can we study that? So if this cancer cell makes, if this cancer tumor, sorry, if this whole tumor with its macrophages, even the macrophages have dual roles. Sometimes they're supporting, sometimes they're not. They decide. But if the collagen, if it is made, then it can stop the tumor spread. All right. So here's your tumor. And if there's a lot of collagen, see this collagen here? See all that collagen here that I'm showing you? Then the tumor will not go to the blood vessel. That will not happen. Okay. So it's good. That means there's no metastasis. Okay, that's fine. So calves, if they made too much collagen, then they will not go to the tumors. But if there's less collagen, then they can go and spread metastasis. Okay. And all of these, the collagen, so this is collagen again, we found that it provides energy, it promotes the tumor. Collagen promotes metastasis. This is what we found also. Too much collagen or more collagen. Then collagen also helps holding the cells together. Okay. And collagen also makes it hard. Okay. It increases the ECM hardness. So how is that beneficial? If it's very hard, the drug cannot go in. Drugs can never go in. They just don't. Because it's making collagen everywhere is making it so hard, making everything hard. So it promotes resistance. So collagen seems to be not a very good thing for uh, for um, for normal cells. More collagen was going to be a problem, okay? Except for one benefit here. So whatever I said is just written in here. I said the same thing. And so if you wanted to treat it, you would break down the collagen, okay? So if you wanted to, it seems that collagen is kind of bad for, so in breast cancers, breast cancer, you can target collagen and you can degrade it. So you can add an enzyme, collagenase, and collagenase will degrade it. And if it degrades it, then probably that it can, then the tumor will be less stiff and then it is accessible to the drug. So, but only one time, only one example, we just found that collagen can suppress cancer. Okay, all right, fine. Maybe it does help to form a barrier and it prevents metastasis. All right, and that's also a different type of collagen, collagen 3. All right, so all good and dandy. All good. So the point is that if collagen is going to increase, we might have a problem. If collagen, then we should assess. All right, how are we going to assess the collagen by histology, and or by cytology? How are we going to do that? Okay, so if we have too much of collagen, then we want to give collagenase enzyme, and we want to break the collagen. That's what we just decided, that too much is bad for you. So collagen can also be a cancer biomarker. So how are we going to measure it by cytology and then use it for some kind of machine learning? Well, people have used something called as, and so this is a very new paper, August 2024, just 1 year. And so collagen in breast cancer, people have used something called as optical histology. Go figure. What does that mean? Well, optical histology is something that is called as SHG fibroscopy. SHG fibroscopy. And hold on, and I forgot what that means. Second harmonic generation. So it's not a direct visualization of the cell, but still it is a direct visualization of the collagen in the tissue. So let's see how that's done. And that's so very soon I can imagine that there might be data on this very soon. There will be a lot of picture data. And then again, for machine learning purposes, this would be for AI, it would be a good analysis to do. All right, so what is SHG fibroscopy and how did they use it in breast cancer? So the collagen content, okay, what's the collagen content again? Where's the collagen? The collagen is outside the cell somewhere. Here, you see this? So here's your cancer cell. The collagen is out here, outside, and it is preventing the immune cell from killing the cancer cell. Okay, it's preventing, it's a barrier. So again, this is collagen here. See, collagen, and well, this collagen is preventing this good T cell from killing this cancer cell. But if you inject collagen A, then it can do that and it can kill the cancer cells. So let's see what happens here. So collagen content, they're not, it's all within the tissues, okay? So it's all, they used cancerous and normal tissue, okay, straight away. All right, and they checked the collagen fibers and they did some kind of a fibroscopy on this. And so this is what it looks like. Okay, so this is what it looks like. So this is what it looks like. It's not really, I have a video, and to tell you what it is, it's not really a cytology, but it is slightly altered cytology. So let's take a look at this video and see what it gives us.
Or second harmonic generation occurs when HistoIndex offers innovative imaging solutions to visualize and quantify fibrosis features in unstained tissues. A unique phenomenon called SHG, or second harmonic generation, occurs when photons interact with a collagen fiber that is non-centrosymmetric in structure to combine into a single photon, bearing its own distinct SHG signal. The SHG signal from the collagen fibers is detected throughout the entire tissue scan. This biophotonic imaging technology provides valuable insights into fibrotic tissues, opening the door to new standards in fibrosis diagnosis that will benefit clinical research
and this was something that when tissue clearing came around and started getting popular, I was wondering if that was really going to cut
Hold
into
on, let
the
me get
amount of
this
two
out of the
photon
way.
microscopy we do here at the facility.
And so the collagen fiber it's not a direct cytology, but you are just getting the collagen fibers from the tissue. And so I imagine that this is going to become, this is fairly new, and it's going to become some kind of a optical, what's the word again? Optical histology. So here it is. These are the what they got for these normal breast tissue for early breast cancer and advanced breast cancer. And this is nothing but collagen content. So it's collagen amounts. And so this was with patients. This was done with patients. You can see that there were different patients here. And what they found is, look at this graph here. What they found is that the collagen area fraction means the collagen content indirectly. The collagen content decreases as the stage in those patients that have higher stage of breast cancer. So overall content density of cancerous tissues from 1 to 4 is significantly lower than the normal group. So they found that there is reduced collagen density. So if there's reduced collagen density in these cancerous tissues, that's kind of like reverse of what we just talked about. Kind of doesn't make sense, but that's how it is. That's what they found. And so the bottom line is that this could be the collagen measurements could be done maybe later on. A lot of indirect understanding could be just from these collagen measurements in the tissue. So it is not really a histology, but it is some kind of mm, optical histology. And I included this in because of the way they said this. And with that, I stop today's talk. Let me just see. I still have 10 minutes to go. So before 10 minutes, I'm ending this talk here. All we just summary what we've learned. We have learned about all the tumor environment cells. There's so many of them. We also learned that ECM is an environment, the extracellular matrix, and that's why collagen came in place. And so we measured collagen by some kind of um, um, histology studies. And then we also looked at various, the fact that you can have tissues and genomic studies together. And also that you can have dual roles of macrophages. They can be either good or bad. They can be pro-tumor or they can be anti-tumor. They can stop the tumor or they can help the tumor grow. And then we looked at angiogenesis and all of the factors related to the TME. Okay, so thank you very much. I will stop it here. So do you have any questions, anyone? Are you there? Do you have any questions? Was there too much that we covered today? Was there just about right? Was it too much of biology? But I think my point is to give you all these ideas so that you can go back and try to see where you can incorporate newer cytology for detection progress.
So I think this is the end of the lecture
Sir, good
session.
afternoon. Good
Oh, okay, Karthik ji. Okay.
afternoon. Sorry to interrupt, sir. Madam, I got a question.
Yes, please.
Is this collagen inhibition therapy still in
Still in what? What was the?
R&D phase, or are
So what they're doing is they're taking the samples from the previous clinical samples, um, and they are examining this. So I think the fact is that collagen has been there. We've known about, collagen for a while, but it was never a major thing to look at. So the fact that these patients, they had these samples, and they could be used for looking at collagen. So the technology is new. It's not like the collagen concept was not there. The theories behind it is there, but it just never has that major importance, only because there are so many other cells to look at. And it was always thought, and it's still thought, and this is fairly new. So this is fairly new as to how to handle it. So most of these studies are trying to say that, the collagen has a dual role. So whenever anything has a dual role, it becomes a little bit hard. It's the same thing for macrophage M1 and M2. For years we're still not able to figure out, can we do anything with it, or can we not do anything with it. But for collagen, I think it depends on the type of cancer. And so, therefore, this is fairly kind of new because of the technology being new, not otherwise. So, um, being able to really assess the collagen amount, that's a big thing. Before, you could not assess collagen amount just by, by looking at, because collagen is already a lot. So you see what I mean? It was there before, it was just never, um, not much was done with it at that time. So I think the optical histology makes a big difference. Does that sound right, Karthik? Uh, and I think that it is much more easier to look for this. Are,
thank you.
are you there?
Thank you.
Uh,
Thank you.
so thank you for asking the question, Karthik ji. And I think this is the lecture 4. Um, that is the ending of the lecture 4 of tumor mm, microenvironment. So next we will we'll start the tutorial 3 uh, presented by Professor Nath, I think in 2:30. After 1 hour, it will start cell-based assay